Hello students, we're going to be talking about chapter 4 today, the cell. So we've already talked about the cell being the basic fundamentals of life. Alright, so here we have a fluorescently labeled cell. We're going to be talking about basically all of this. So the blue part is the nucleus. You can see the filaments kind of going everywhere. That's the green parts, and you see the red parts there. Those are actin filaments. All right, next slide. So how do we discover what cells are? First of all, these are kind of the people that discovered how cells came about. So first, Robert Hooke looked through a cork at a microscope. He called them cellulae. This is his microscope. It was kind of primitive. He looked at it, little small holes inside of those corks. A little bit later, Anton van Leeuwenhoek, don't really know how to pronounce his name, but you know, discovered tiny animalcules, protists, bacteria. He also looked at sperm cells. That is his microscope. It's again kind of primitive, but you can kind of see little things on that. There's the plate, and then there's a little hole where that plate is, and you can kind of see through it. And then the specimen would be a little pin, and then you would see through a lens in that hole, and you would see it. About the 1820s, Robert Brown, with a more advanced microscope, could see the nuclear bodies within the cell. He wasn't really able to see organelles yet, because they didn't invent the staining process until later. But the nuclear body was a little brown circle, so you're able to see that without a stain. This is the cell theory that they came up with. So this is the basis of cell theory. All living organisms are made up of cells. They have to have one or more cells. And they can only arise from pre-existing cells. Basic structure and functions of cells. Cells carry out all essential processes of life. All cells contain DNA or RNA, hereditary information that directs molecules to do work. Right? They undergo homeostasis, respond to the environment, and reproduce to pass on heritable traits. Right? Basically, the functions of life. Cells contain DNA, undergo homeostasis, respond to the environment, and reproduce. Right? Okay. So how do we see cells? This is through microscopy. They're too small to see. Most cells are too small to see, right? Some cells, there's very small amount of cells that you can see. They're really, really big. But most cells you have to look through a microscope. There's different types of microscopes that you can use. There's a light microscope that you would be able to use in lab if we'd been there. That's off to the side here. You can look through it and you can see bacteria, you can see small little protists or whatever swimming around. And then there's an electron microscope. Electron microscopes, instead of the light microscope that just uses light, electron microscopes actually bounce electrons off of the specimen. Most of the time, electron microscopes, you have to coat the specimen in gold or some type of really expensive plating. Otherwise, it'll actually burn the specimen. But in that case, the electron microscope actually has atomic levels of resolution. So, depending on what you're looking at, you can actually bounce electrons off of, like, what is it? You can bounce electrons off of pollen grains, or you can bounce electrons off of the very smallest, what was that called? The smallest snowman. They built the smallest snowman out of atoms. You should go see that. Go look it up. Magnification versus resolution. In a microscope, there's a difference between magnification and resolution. You may know this difference, but I'm still going to go over it. So magnification is the ratio of an object as viewed to its real size. So like 4x, 10x, or 40x, right? 4 times its regular size, 10x times its regular size, or 40x times its regular size. As opposed to resolution, that's the minimum distance between that you can see 2 different objects. So resolution, a long time ago, Galileo used to be able to see Saturn. And what he saw was Saturn and 2 moons. What he thought he saw was Saturn and then like 2 different moons on either side. But what was actually going on was that there was the ring. He didn't know that the ring was actually the 2 moons that he saw. He thought there was 2 moons on either side, but it was actually the ring. So what his telescope was seeing was actually not what was going on. So he wasn't getting the right resolution with his primitive telescope. All right, size limitations. So cells are limited in size, right? You can't have organelles just bursting at the seams. So surface area determines the amount of substance that can travel between the membranes of the cell. So you want to have a lot of surface area, but you don't want to have a lot of volume. Right? This is kind of what I was talking about before with mitochondria. So you can use folds inside of those membranes to increase the surface area, but not increase the volume. So you can see the mitochondria on the side that shows those folds, those cristae. So it increases the surface area. You can also see the endoplasmic reticulum. It has a lot of folds in there. It increases that surface area, but it doesn't increase the volume within the cell. All right, plasma membrane. Kind of talked about this one before. Phospholipid bilayer, right? It has a hydrophilic head, hydrophobic tail. It also holds important transport proteins, right? To transport between that membrane layer. Otherwise, it would just be a wall. You need to be able to transport between the outside and the inside of the cell, but it's selective. It only selects certain things to transport between that phospholipid bilayer. Okay. Selective transport of ions and water-soluble molecules. Inside the cell, right, we have the DNA, cytoplasm. The cytoplasm is the movable liquid inside the plasma membrane. It's just basically everything inside the plasma membrane. It contains the cytosol, which is just the solution, right? Including organic molecules, ions, and organelles. Cytoskeleton, those are strong proteins of structure, so it structures the cell, right? We'll talk about that in a minute. Maintains the cell shape, right? And plays a role in cellular division. Right, so we have two different domains of cells. Prokaryotic, that doesn't have the nucleus, but it has a nucleoid region. It's not bound by a membrane, but it's in the center of the cell. Doesn't really have internal membranes. Archaeans did split off from bacteria, and do share some features with eukaryotes, but it does have its own domain branch. Okay, and the eukaryotic cells, these do have a true nucleus because it has a nuclear membrane, right? And it has membrane-bound organelles. More to say about prokaryotic cells. Most of the time, prokaryotic cells have three-dimensional shapes. These are spherical, rod-like, and spiral. Spiral are like those spiral Cheetos. You ever had those spiral puff Cheetos? Those are like that. If you don't know what I'm talking about, kind of look them up. You know what they are. For most species, the DNA is circular. So there's single circular molecule. That's the prokaryotic chromosome. So instead of ours that's linear, theirs actually goes in a circle and it binds together in a circle. It's kind of weird, but it actually does bind together in a circle. The plasma membrane is surrounded by a cell wall. It's called a glycocalyx. So prokaryotic cells have a cell wall all the way around it, and it depends on how the cell wall is structured. So bacteria usually have what's called gram-negative or gram-positive. So it'll depend on where the cell wall is as to where it's gram-positive or gram-negative. If it's gram-positive, the cell wall is on the outside. If it's gram-negative, the cell wall is on the inside of a plasma membrane, and it's a lot thinner. All right. When the glycocalyx is loosely associated with the cell, it's a slime layer. It's a capsule, so it's kind of what I was talking about. Flagellum. So this is kind of the way that bacteria move. There's either flagella or there's cilia. The flagella are little tails. They usually spin around and they kind of have this corkscrew effect. So it just rotates in a socket, pushes this cell through liquid media. Pili are kind of like mating organs. So, not really organs, but hairs, if you will. What they'll do is they'll just kind of join pili, and then they'll exchange some genetic information, and then kind of go their separate ways, and then they'll split. Then we talk about eukaryotic cells. Of course, these have a true nucleus, membrane-bound organelles. Cytosol participates in energy metabolism. That just means that the proteins inside of it do break down molecules and other things. All right. Cell fractionation. Not much to say about that other than there's whole cells. You can put it into a gravity blender, and then you can separate the cells based upon their organelles or substances inside the cell. Mainly because the organelles are differentiated, you can separate the differences using cell fractionation. All right, the eukaryotic plasma membrane carries out various functions because of those embedded proteins. So you have channel proteins that transport substances in and out, receptor proteins that can bind to the environment around it, and then you can have immune system proteins that can recognize different other things. So like your T cells or your white blood cells, right? So your T cells go out and they can recognize other things as foreign, and it'll attach to those, and then your white blood cells can attach to the T cells, and attack. Supportive cell walls, right, surrounds plasma membranes of fungal, plant, and protist cells. So we'll talk about the cell walls in a bit. The nucleus. So, nuclear membranes, nuclear envelopes. You can say it either way. Consists of two membranes. We have the nuclear pores, right? What comes out of those nuclear pores? The RNA, right? The RNA and a certain protein. There's some other things that can come out too, but mainly the mRNA and a protein. All right. So those go off to the rough endoplasmic reticulum, or a ribosome, and those get translated right, into proteins. We have some nuclear proteins, nuclear localizing signals. Don't really need to know about this part. It's an interesting part, but it's not really that important. The liquid within the nucleus is called the nucleoplasm, right? It's plasm within the nucleus. The nucleus contains chromatin. Chromatin is DNA with proteins. So DNA in eukaryotes is linear. That's the eukaryotic chromosome, right? If it's wound up together, spun and wound up together, it's a chromosome. Usually it contains one or more nuclei. It's basically those genes coding for RNA. Nucleoli, excuse me. It's the genes, it's the coding genes. What do I mean by that? There are non-coding genes. Anything that doesn't need to be coded in a specific region. So if something's coding for my ear up here, it doesn't need to code for my foot, right? So nucleoli up here is for my ear. Does that make sense? All right. Expression of genes is carefully controlled according to the function of each cell, right? That's just what I was talking about. Okay, next one, ribosomes. Ribosomes consist of a large and small subunit. So you have the large on the top, at least in this picture, large on the top and the small one on the bottom, right? Gets that mRNA through between each of them. And then it starts to code and makes the amino acid chain. All right, some float around the cytosol, others attach to the rough endoplasmic reticulum. Any attached to the ER goes through the special path to follow the other organelles down the cell, usually going through the Golgi apparatus. So we just talked about the rough ER. Let's just talk about those. So endoplasmic reticulum, it's the extensive interconnected networks. Endoplasmic reticulum are like factories, right? These are big, extended pathways, right? Let's say that there's a lot of substances just floating around the cell. If you needed to get something, wouldn't it be better to get something within a room rather than in a town? Right? So you want to get something within a close distance rather than a large distance. Right? Because it's easier to get to, it's faster to get to it. Right? That's what the ER is doing. It's keeping ions, keeping molecules within a close distance so that it's faster to get to it, it's easier to get to it. So the rough ER is a main site for protein synthesis, and a smooth ER is the main site for lipid synthesis. Lipids to make the membranes, lipids to make, to fix things. Okay, The rough ER is rough because the ribosome is attached to the membrane. So the Golgi apparatus is the sac of flattened membranous, it's a sac of flattened membranous cisternae. Proteins made in the ER enter the complex, basically delivered by transport vesicles. These vesicles are just basically little boxes of membranes. Proteins are chemically modified by removing segments or adding functional groups, lipids, carbohydrates, whatever, right? So in this case, if you think of the ER like a factory, you can think of the Golgi complex as like Amazon, right? This is making it, this is shipping it out. So the modified proteins exit through vesicles that butt off from the membrane. So these are exited through the vesicles like boxes. And they just kind of go off. So we go into the next slide. So the exocytosis, they can either exit through secretory vesicles through exocytosis. Basically, the Golgi complex tags the protein for their final destination. Fly. And they can go outside the cell. All right, next slide. And sometimes, if it comes from another cell, it can come in through endocytosis. So it's just coming in to the cell. And that's called an endocytotic cell, vesicle. Lysosomes, small membrane-bound vesicles containing enzymes that digest molecules. In a process known as phagocytosis. Phago meaning eating, cytosis meaning cell. Or cyto meaning cell, cis meaning process of. So eating, process of eating a cell. So hydrolytic enzymes, hydrolytic meaning water eating, or water splitting, excuse me. Remember lysis, hydrolysis. Hydro meaning water, lysis meaning splitting. So phagocytosis is cell, process of eating a cell. Hydrolysis meaning splitting of water, or water splitting. Okay. pH within the lysosome is usually very acidic. Okay, that just means that it's able to split, it's able to disintegrate that whatever it's eating. It's formed by budding of the Golgi complex, and it's usually found in animals, not really in plants, because they have their own process. Usually they just kill off whatever they don't need and just kill it. These can kill off anything from the mitochondria to an entire cell. Speaking of mitochondria, cellular respiration undergoes in the mitochondria. We should know that. So, all things needed for cellular respiration, we need food, we need water, and we need to breathe, right? We need oxygen, obviously. In order to go through cellular respiration, we don't undergo fermentation, right? We can't use the alcohol. That'd be interesting if we did. Right, so we need our lungs, we need our digestive system in order to undergo cellular respiration through our mitochondria. Right, so here's the mitochondria over here. It has two membranes. The outer membrane covers the whole organelle. The inner membrane has those cristae, right? We talked about the folds and everything. Both membranes surround the matrix on the inside that contains the DNA, ribosomes, other components that's needed to undergo its functions. And what does the mitochondria produce through cellular respiration? It has to produce ATP. That's the energy producing compound that's needed through life. So in the beginning, how did mitochondria get introduced into the cell? Because it has a double membrane and it's not really a part of the system. So the endosymbiotic theory is one of the first theories that was proposed to say how mitochondria and chloroplasts might have been involved. Basically, there was a mutual relationship between an ingested prokaryotic cell and the eukaryotic cell that ingested it. So there are some problems to this theory. It doesn't really occur naturally. Like, you can't just find this right now in the wild. You can't just see this happening right now. It can't be tested. It can't be tested again, right? It's not a testable theory. So there's another theory, it's called Bleb's theory, where basically there was a eukaryotic cell that had what's called blebs, little arms that may have stuck out at some point. And they may have just grabbed these prokaryotic cells, and over time they just kind of intertwined and they just kind of overtook these prokaryotic cells. That doesn't really make a whole lot of sense either, but I may not be explaining it right either. If you want to go into a little bit more detail on the Bleb's theory, it does make a lot more sense than the endosymbiotic theory, but that's up to you. So within the cells, there's the cytoskeleton, right? We talked about the cytoskeleton being its main structures. interconnected system of protein fibers and tubes that extend through the cytoplasm. So if you see that at the bottom, you see all those little tubes that are just kind of branching out. These play an important role in the function of movement of the cell. It plays an important role in the shape of the cell, and especially in plant cells. It plays an important role in its structure. Microtubules. The walls of the microtubules consist of 13 protein filaments. So, the dimers are organized head to tail in each filament, giving the microtubule a polarity. You have the positive and the negative end. They can change their length depending on the addition and the removal of the tubulin dimers. So you can basically change the length depending on how many proteins you want within that microtubule. These are eukaryotic motor proteins right here. They can push or pull against the microtubules, and they will walk on the microtubules to travel. They can basically pull large organelles and just travel along those microtubules like highways. So two of them are called kinesin and dynesin. And they'll just go backwards of each other, like that. Microfilaments, these are smaller. They're thin protein fibers assembled from actin subunits. So they also have polarity. They have structure and locomotor functions. These are the ones in your muscle fibers. Or not the ones, but like the ones in your muscle fibers are microfilaments. The ones involved in cytoplasmic streaming. So in a plant, cytoplasmic streaming, you'll see the chloroplast movement throughout the cell. Divide the cytoplasm when animal cells divide. So when your cells divide, microfilaments are helping. And then there's some intermediate filaments that are between microtubules and microfilaments. So there's just some intermediate filaments that have some functions, but we don't need to go into it. All right, next slide. Flagella and cilia. So we kind of talked about flagella a little bit. Whenever we talk about flagella, these are long motile structures that extend from the cell surface. So, you know, that tail. They propel a cell through watery medium. The main structure of a flagella is a circle of 9 double microtubules surrounded by a central pair of single microtubules forming a 9+2 complex. That's confusing. If you look at the diagram above, you'll be able to see it a little bit more clearly. So there's this really complex structure of microtubules, and that's what's going on inside the flagella. Also, the cilia kind of have that similar structure in each one of their tiny little hairs. And that's at the bottom there. So cilia are little hairs that kind of waft the liquid around them. All right, plastids. Plastids are found in plants. Chloroplasts, as we talked about, are yellow-green, site of photosynthesis, right? Ameloplasts, colorless, they store starch. Chromoplasts, they're red or yellow. They also store some sugars in them. They're responsible for the colors of like autumn leaves, also fruits. All plastids contain DNA and code genes. So chloroplasts, they are surrounded by two membranes. Remember that endosymbiosis theory. Whenever it came in, it got another membrane around it. It completely covers the stroma. So, the stroma consists of flattened enclosed sacs called thylakoids. Many thylakoids on top of each other are called grana. Thylakoid membranes contain chlorophyll. So one stack is called, one thing is called a thylakoid, the stack is called a grana, the liquid surrounding it is called the stroma, and then you have the two membranes around it. In all cells, you have a central vacuole, but in plant cells, you have a really big central vacuole. And there's a reason why. Plant cells need what's called turgor pressure. If you've ever watered a plant and then seen it like come up and then not watered a plant for a long time and seen it wither, then try to water it again, then it comes back up again. That's the main reason that the central vacuole is important. The central vacuole keeps that turgor pressure in each cell so that it can stay upright. So it's like a balloon in a very tight box. So each one of these has about 90% of its volume inside of that cell covered by that vacuole. The central vacuole is surrounded by a tonoplast. Okay, so the balloon is called a tonoplast. Inside, what it is inside is called a cell sap. Okay, if you were to pop the balloon, it would be cell sap. Functions of the vacuole. So all of that cell sap inside is mostly salts, organic acids, some waste product, sugar. There's also some pigments inside. If you have any type of purple plant or some type of bluish flower, what's called anthocyanins is inside, and that's going to be inside that vacuole. So it contains enzymes that break down biological molecules. Sometimes it'll contain some type of acid inside of it. Sometimes it will contain a poison, depending on where it is inside the plant. Cell walls. So we all know that plants have cell walls. There's also bacteria that have cell walls. So gram-positive versus gram-negative. Over on the side here, we see that gram-positive has a very thick cell wall on the top, versus gram-negative has that thin cell wall in between two plasma membranes. So you can see kind of the difference there. Primary cell wall versus secondary cell wall. There is a difference between primary and secondary in plants. So secondary cell wall, if you build a lot of secondary cell walls, that translates to what's called wood. Inside of a plant, lignin reinforces that wood. It just makes it really hard. Consists of cellulose fibers, so you can't, eat it. Between each cell wall, there's what's known as a plasmodesmata. So whenever a plant gets water, it can transport that water between each cell, or minerals, or whatever it needs to transport between. But it's selective, right? So the ER, the endoplasmic reticulum, goes between each cell. There's what's called a middle lamella, made up of polysaccharides, pectin, between each cell wall. So you have one cell wall here, one cell wall here, and in between them is that middle lamella, right? So one cell up here, one cell down here, one cell wall, middle lamella, right? And then you have, in between that, is the plasmodesmata. And you'll see them running, an ER running between that. So we talk about the animal cell surface. Not really much to talk about here. There's cell adhesion molecules that bind cells together. Certain ones that can bind them together, like your skin. You don't want to allow certain things inside your skin, obviously. So there's certain molecules that can bind your skin together. Cell junctions that seal the spaces between the cells. And then you have the extracellular matrix that supports and protects cells, provides mechanical linkages between tissues. Okay, for your attendance assignment, I want you to tell me one transport protein inside the plasma membrane. Just look up one and then put it on Blackboard. All right? And I will see you in lab.